Hello everyone and welcome back to my channel. Today we will be talking about different types of cell signaling and see an example of how a signal molecule can start a chain reaction that can ultimately lead to a gene being transcribed. So there are 4 types of signaling. The first is endocrine signaling, which involves a signal cell secreting a hormone. This hormone has to travel far distances, and to do this it enters the bloodstream and exits once it's found its target cell. An easy way to remember this is by recalling that the endocrine system is responsible for synthesizing hormones. Some examples of signaling molecules for endocrine signaling include testosterone and estrogen. Now, let's say you're standing close to your friends and you want to get their attention. You can simply tap their shoulder to send a signal. And guess what? Paracrine signaling works the same way. In paracrine signaling, it is the complete opposite of endocrine, and it instead works over short distances through diffusion. Our third type of signaling is neuronal signaling. As the name suggests, it involves neurons. After a signal molecule, in this case a neurotransmitter, travels down the neuron's axon, it reaches a chasm between the neuron and the target cell called a synapse. The neurotransmitters then get secreted onto receptors on the target cell. Finally, contact-dependent cell signaling requires direct contact for a signal molecule to bind to a receptor on the target cell. And this kind of cell signaling is very important for cell differentiation. Well, you're probably wondering what happens after a signal molecule binds to a receptor. Well, do I have a great pathway for you. But before we dive in, there are some terms you should know, such as secondary messengers. So, a secondary messenger is a molecule that relays signals received at receptors on the cell surface. A protein kinase is an enzyme that adds a phosphate group to a substance. A transcription factor is a type of protein that regulates the synthesis of RNA from DNA during transcription by binding to a specific DNA sequence. ATP is the energy of a cell, also known as adenosine triphosphate. A transmembrane protein is a protein that is embedded in the plasma membrane. And adenylyl cyclase is an enzyme that makes cyclic AMP. Now that we have those terms out of the way, let's get into our pathway. So, G protein-coupled receptors are transmembrane proteins found in plasma membranes. After a signal molecule binds to its receptor, the GPCR, or the G-coupled protein receptor, will seek out a G protein. Fun fact, G proteins are called heterotrimeric proteins because they are composed of 3 subunits, an alpha, beta, and gamma subunit. But for our purposes, the alpha subunit does all the heavy lifting. After a GPCR binds to a G protein, the G protein becomes active, and GDP gets swapped out for GTP. If you don't have GTP binded to the alpha subunit, your G protein will stay inactive. The active G protein then has the alpha subunit disassociate from the beta and gamma subunits, and it makes its way towards another enzyme called adenylyl cyclase. Adenylyl cyclase will cause ATP to become cyclic AMP, a secondary messenger. Cyclic AMP amplifies the original signal and activates protein kinase A, which will then phosphorylate transcription factors and allow for transcription of a particular gene. Ta-da! Now you know the importance of signaling molecules and what happens behind the scenes after it binds to a receptor. Anyway, before we end this video, let's do a quick recap of the GPCR pathway. So again, you have a signal molecule, binds to GPCR, and it's going to activate a G protein, but before your G protein can become active, you need to switch out GDP for GTP. Then your alpha subunit disassociates and goes to search for adenylyl cyclase. Adenylyl cyclase, when it's active, turns ATP into cyclic AMP, and cyclic AMP will activate protein kinase A. Once protein kinase A is active, it will go phosphorylate a transcription regulator, allowing for transcription to occur. Anyway, a big credit to Essential Cell Biology 6th edition, because that's where I got all of my photos, and it really shows the pathways very accurately.